Okay, so today's lecture is on aerobic respiration and going through the chemical pathways that are involved in that process. For our objectives here, we have three simple ones. Remember, when you get to the end of the video lecture, you want to go back to these objectives, see if you can answer them. If not, you may want to watch the video again, or review your notes. As always, if you have any questions, feel free to ask your teacher by email or in class. So, what are we talking about? Overall, in respiration, we take glucose and we break it down into energy that we will use in our cells. And basically, we are going to be making ATP molecules. And what you see down here in this corner, this is an ATP molecule. Remember that it is part of the nucleic acid macromolecules. It has an adenine here, a ribose, and it has 3 phosphate groups on there. And remember, when we are ripping off one of these phosphates, that's how we release energy, and it breaks the ATP down into ADP. Then when we receive energy, like from glucose, we're able to store it in those ATP molecules until we need to use them for certain processes. Where is this going to be happening? Well, the mitochondria is the main star when we talk about respiration. We've already discussed structure and function with the mitochondria in our first video from this unit. So just pointing out again, remember the mitochondria have double membranes. They have this outer membrane, which is the darker brown color, and then they have this inner membrane, and it's folded in here so that there is a lot more surface area on this membrane than the outer one. And remember, those folds are called the cristae. So then we want to introduce two other spaces. The blue space, which is the complete interior of the mitochondria, that is known as the matrix, that is the very center. And the intermembrane space would be what is between the outer membrane and the inner membrane. Okay, so the matrix and the intermembrane space are both spaces that are either within the inner membrane or between the inner and the outer membrane. So here is a flowchart overall of what is going to happen, and I just want to annotate this, and I think it's important for you to as well. Okay, so we're going to start with a process known as glycolysis, and glycolysis is the first breakdown of glucose, so the first step in the chain. But then we have two different options, and the options here are going to depend on the availability of oxygen. Okay? If we have oxygen available, then what we're going to continue with is aerobic respiration, and that's going to go inside the mitochondria, and that's going to take us through the Krebs cycle and the electron transport chain. Okay, so this side is what we call aerobic respiration. All right, and both of these here, they're going to take place in our mitochondria. Okay? But when oxygen is not available, we need to have a second option. And so that option is known as fermentation, and this is what we would call the anaerobic process. And it literally means without oxygen. So you've probably heard of this word aerobic before when you think about aerobic exercise. And if you think about all the things like jumping jacks and leg lifts and, kicking and, arm swinging, what is it all doing? It's causing your heart rate to elevate. And what is the purpose of your heart? It pumps your blood, which is circulating oxygen and nutrients around your body. So aerobic respiration uses oxygen, and the rest of the steps take place in the mitochondria. But when oxygen is not available, then we're going to talk about going through a pathway known as fermentation. This is going to be in a follow-up video lecture. Today, what we're doing is we are focusing only on aerobic respiration. It's important to note both have glycolysis as the first step. Okay? All right, let's get rid of that. All right, so the first step, as we said, is going to be called glycolysis, and this happens in the cytoplasm. So we're not inside the mitochondria. And that's why glycolysis is able to happen in both aerobic and anaerobic respiration. Okay, it does not require oxygen, but it also does not exclude itself from the aerobic pathway. It is the first step. Down here in the bottom is a little graphic. This shows you our overall picture of what's going to happen in aerobic respiration. Okay. If we literally break down the word glycolysis, lysis meaning to break down or split apart, glycose is referring to sweet. So what are we doing? We are breaking down glucose. As I said before, glycolysis does not require oxygen, but it needs some energy. So if we need energy to get this process started, which is a cellular chemical reaction, where do you think we're going to get that energy from? We're going to have to use a little ATP. Okay, we need to use some ATP molecules for that. So, in order to get out some energy, you need to invest some energy. So we call these the investment and the payoff phases of glycolysis. So here we see the glucose molecule, 1, 2, 3, 4, 5, 6 carbon, right? That's representing glucose. And we're going to break it down into 2, 3 carbon molecules. And these molecules are called pyruvic acid, or they're also called pyruvate. Let me put that in there for you. Pyruvate. They mean the same thing. Okay. So that's all we're doing. We're basically taking the glucose molecule and breaking it in half. Okay. And when we break it in half, it becomes, these two other products, these pyruvate. But in order to do that breaking, okay, remember we talked about the breaking of bonds and the reforming of bonds. There's going to be an energy investment. So ATP is readily available within our cells, so we're going to spend 2 ATP, right? So it's sort of like we subtract 2 from our overall total. But during this process, okay, when we reform those bonds, what happens? We get a release of energy. And how many have we released? We have released 4. So if we do some quick simple math here, -2 plus 4 means overall we have a net gain of 2 ATP molecules. And that's what it says down here, a net gain of 2 ATP molecules. But then we also have these guys in pink. These are NADH molecules. And you should be thinking, that kind of sounds familiar. We talked about an NADPH molecule when we did photosynthesis. Well, this is its close cousin, and guess what it does? It is an electron carrier. Okay? So just like in photosynthesis where we had some systems, where we had some systems that allowed us to extract electrons that were released from a chemical reaction and then trap them in molecules, we're going to use the same thing over again in respiration. I guess the saying goes, if it ain't broke, don't fix it. This is just another type of chemical reaction used to extract and transfer energy. So why not use a similar pathway to what we've used before? So we have these NADH molecules, or we'll just call them electron carriers in general. Okay, let me get rid of that. So, check 1. We've gone through glycolysis, all right? Now, again, as we said, we're going to assume we have oxygen readily available to us. So we're now going to move into the mitochondria for step 2, the Krebs cycle. And that is going to happen specifically within the matrix. So the very, very, very center of the mitochondria. We have to go through the outer membrane and go through the inner membrane, going into the very center of the matrix of the mitochondria. Okay. And what happens here? Well, it is a cycle, and be careful that you don't get it confused with the Calvin cycle or the dark reactions from photosynthesis. A lot of times students mix those up. Again, just like in photosynthesis, there is a lot of stuff going on here. Many, many different chemical reactions that are occurring, that are, using energy, creating energy, and other products. But let's just talk about what's gone in and what is coming out. Okay? What's most important, what went in, is that pyruvic acid molecule. Okay? See the 3 carbon up here? That pyruvate or pyruvic acid molecule goes in. Okay? And as we crank ourselves through the cycle, we're going to make some products. So here's where we make 3 of our carbon dioxide molecules. We're going to make 1 ATP molecule. And here we see our NADH. We make 4 of those. And FADH2. Again, it sounds really similar to the other 2 electron carriers. So make a note of it. Yes, both of these are electron carriers. They're just different types. Okay, so in cell respiration, we have not only NADH, which can carry electrons, but also this FADH2 molecule that's able, that's able to carry electrons. Okay, but is this the end result? No. Okay, because if I remember my equation properly, C6H12O6 plus 6O2 yields 6 H2O molecules. Okay, so where are my other 3 molecules coming from? What's going on with that? We have to double the order. Remember, at the end of glycolysis, we made 2 pyruvic acid molecules. And that Krebs cycle only looked at what the results were for 1 of them. So what ends up happening is you essentially have, here, let me just do this. We have 2 pyruvic acid molecules. Oops. And we're going to have 1 go this way and 1 go this way, and it's going to go through the Krebs cycle once over here, and it's going to go through the Krebs cycle over here. So we have to double the order because there are 2 pyruvic acid molecules. So these are now our totals. These are the numbers I want you to remember. You do not need to know the 3, 1, 1, and 4. 6 CO2 molecules and the 2 ATP molecules. We mentioned this before. You do not need to keep the totals on how many electron carriers are made, but just note that there are significantly more made here than were made in glycolysis. Okay? So, what's going to happen? We broke down the glucose molecule, and as we continued ripping it apart, we took the 6 carbons from the original glucose, and they wound up in the 6 CO2 molecules that were made as a product from the Krebs cycle. So that is released back out into the atmosphere when we exhale. That's how we exchange our gases through our lungs. The ATP that we make here, that's going to be used to supply energy for the cell. We're not making a whole lot, right? Glycolysis, we had a net amount of 2 ATP. Here, we have 2 ATP made as well. It doesn't really sound like the big amount of ATP that we need to make. So I have a feeling there's another step involved where we're going to have a boatload of it. And then the last thing from Krebs cycle is we made more of these electron carriers. Where are we going with them? We're going to an electron transport chain, and that is going to be our next and our last step in cell respiration. So if you're following along, we went through glycolysis where we broke glucose down into pyruvic acid, made a few electron carriers and a few ATP. Then we moved that pyruvate into the Krebs cycle where we were able to extract more energy from it. We made more electron carriers, a little bit of ATP, and some carbon dioxide as waste. Now we are at the electron transport chain. Whereas the Krebs cycle happened deep within the matrix, the electron transport chain is going to happen across the inner membrane. We're going to literally be in the membrane. Okay? This should probably look familiar. Again, if it's not broke, don't fix it. In the light reactions, we saw a similar electron transport chain, but be careful because the details are different in how each organelle uses it. Here, what is going on, we have our intermembrane space here. This is the inner membrane, and then this is the matrix on this side of it. Remember, the matrix is where the Krebs cycle happened. So as the products from the Krebs cycle are coming off, that's our NADH and our FADH2. These are those electron carriers. Well, now we're ready to use the energy from them. So we're going to break the electron carriers down so they release their electrons. You see how we make almost like this little roller coaster of electrons? We're going to bounce these electrons through these proteins in the cell membrane. And remember, electrons carry energy, so as they emit this energy, they're going to allow reactions to happen. The main thing that's happening here is they can actively push hydrogen ions across the membrane. And then what's going to happen after that is that the hydrogen ions will naturally diffuse back across the membrane. So remember, they're going from low to high concentration actively, being pushed by the energy released from these electrons. But then when they're in a high concentration here, they naturally are going to diffuse back across the cell membrane. And we'll catch up with this part in a second here. What has happened to these electrons? Well, we've zapped them of all their energy. They are now tired and worn out. And here we're going to take in the oxygen. This is where we use it. We team it up with 4 electrons and 4 hydrogen ions, and we're going to produce 2 water molecules, and these are given off as a product of the electron transport chain. And then the last thing here that I wanted to focus on again, I said we'll come back to it. These hydrogen ions, as they move across, okay, their movement generates enough energy to turn ADP into ATP. And this is where our main focus is. Okay, so again, those protons or those H plus ions, they're naturally going to diffuse back through that protein channel that's embedded in the membrane. And as they do so, there is an enzyme sitting on the end. You do not need to know the name of this enzyme. I'm just explaining it to you for better understanding. But this ATP synthase molecule is going to be an enzyme and is going to facilitate the ability to turn ADP into ATP. Remember, we need to add that phosphate group on, and when we form that bond, it makes ATP, and now we're able to store energy in the molecule. Very important thing to note that the hydrogen ion does not go into the ATP molecule. It simply passes through, and as it whizzes on by, its energy from that movement is able to convert ADP into ATP. Alright, so let's look at the big picture here. And the most important thing that I want you to get from this will be our ATP totals. So glycolysis is happening in the cytoplasm. Overall, as a net, we make 2 ATP. From the Krebs cycle, we also made 2 ATP. Remember, we're going to go through that cycle 2 times, one for each of those pyruvic acids that we get out of here. And the electron transport chain at the end, once we have gotten all of our electrons carried into the transport chain, we're going to make 32 ATP molecules. So, add them up, and what is the big total? 36 net ATP. This is the number you need to know. 36 ATP molecules are what you are able to extract from the energy stored in one glucose molecule. Okay, that is the amount of energy you get from one glucose molecule. And when you think about the millions upon millions of glucose molecules that you are ingesting on a regular basis, as your body breaks them down within cells, you are able to release that energy, convert it into cellular energy, which can be used to power protein synthesis, active transport, cell division, any enzyme reaction, any chemical reaction, anything that the cell requires energy for is going to be powered by ATP. And this is why it is so very important. So we've talked a lot about graphic organizers, and as a literacy component to the course, I'm trying to model as many of these as I can for you. What I want you to do is pause the video here, and I want you to see if you can fill in this table on your own. Please do it in pencil, okay? And then I will go over the reactants and products with you. So pause here, and then after you have filled this in, I will go over the answers with you. Okay, so let's take a look at what we have going on here. In glycolysis, oops, sorry, let me get this down here. Okay, in glycolysis, what do we have going in? We have a glucose molecule that is going in, and we also need to invest some ATP. Okay, and we'll just keep track of the ATP counts on the product side here. We don't need to keep track of anything else. All right. So we have the glucose going in. We're going to break it down, and that's going to yield some pyruvate molecules. We're going to make 4 ATP, but overall, because we spent 2, we're going to make 2 net ATP. Oops, that's a T. And we're also going to make some electron carriers. Okay. Those electron carriers are going to move down into the transport chain. ATP is going to be used to power things, and the pyruvate is going to move on into the Krebs cycle. So the pyruvate becomes our reactant over here, and that's the one I would focus on. From the pyruvate, we make some CO2 molecules to give off, and remember from our overall equation we know we got 6 there. We're also going to make another 2 ATP molecules, and we make several more electron carriers. These are those NADH molecules and the FADH2 molecules. When we get to the transport chain, we're going to need to use oxygen, and we're going to need to use those electron carriers. Okay. And what do we make? We make 32 ATP molecules, and we also produce water as a byproduct. Okay? Remember, the oxygen teams up with some of the hydrogen ions and the leftover electrons, and they release water molecules. So this is your overall. Remember, important notes: 2 plus 2 plus 32. We're looking at 36 net ATP, and this is the total for 1 glucose molecule. Okay, so this is a good example of a graphic organizer for you. So that's the end of aerobic respiration. The next online video is going to look at the pathway that we go through when oxygen is not available. And we're going to talk about some different examples of different types of organisms, including yourself, that are going to use that pathway. Go back, check out the objectives. If you have any questions, again, review your notes or contact your teacher.